fantastic. It is a distinct honor to be here today to introduce to you our distinguished visitor, Dr. Eduardo Sontag. He's visiting us this week as the 2025 Polyak-Łojasiewicz Distinguished Professor. And today he will give us the first lecture, and then there will be a second one on Friday. So we will have also a social event. We'll discuss. For right now, let me just quickly give you a brief introduction into the manifold achievements by Eduardo. He obtained his degree in mathematics from the University of Buenos Aires in 1972. On that same year, he published a book on themes in artificial intelligence. So 1972, in case you guys were already doing math at that time. Got his PhD in 77 at the University of Florida under the supervision of Rudolf Kallmann. And then from 77 until 2017, he was a distinguished professor at Rutgers University and was a leader in a number of institutes and centers at Rutgers. Since 2018, he's been a distinguished professor at Northeastern with a number of affiliations, bioengineering, mathematics, chemical engineering. And then he also has a position at MIT in the Leeds Department, the famous Laboratory for Information Decision Systems, as well as he's a faculty member in therapeutic science at Harvard Medical Schools. So the question is more like, what is he not affiliated with? So he has research interests spanning several areas of controlling dynamical systems, including systems theory, systems molecular biology, cancer and immunology, machine learning, and computational biology. I believe he'll talk to us about many of these themes today. He's published well over 500 between journal papers, monographs, book chapters, including, as I mentioned, an early work on artificial intelligence. He's a fellow of multiple societies, and he has been recognized by, he received the Wright Prize in Mathematics from Sion, the Bodhi Lecture Prize from IEEE CSS, the Control System Fields Award from IEEE, the Richard Bellman Control Heritage Award from A2C², and a couple of IFAC awards just in case. So basically every professional society in our area has given him the most distinguished award. As recently as last year, he was induced into the American Academy of Arts and Sciences. So it is truly a distinguished honor to have Eduardo with us this week. I hope you had a chance to interact with him and attend both today and on Friday. And with that, Eduardo.
Thank you, Francesco. Yeah, thanks. So after that, now it goes downhill, okay? So just, it's horrible to set expectations. But anyway, no, I'm so delighted to be here because really, I don't know, was anybody here when I was here about 20 years ago and 22 years ago? We had, they called it the Sontag Hour that Peter organized. So you were here, okay. And it was so much fun. We met every week to discuss problems in control theory. And it was so much fun. So, and of course, Peter did an amazing job setting this up and bringing this brilliant group of people. I consider Santa Barbara to be the center for control theory in some sense. It's just amazing. Anyways, so let me just, so as I always say, fasten your seatbelts because I'm going to talk pretty fast and I'm going to accelerate towards the end. And I'll have like 20 different lectures in one. So, you know how I am. So anyway, but I'm here for the whole week. So anybody wants to talk to me later, say, hey, that went too fast. Why don't you tell me more about something? I'll be very happy to, have a separate meeting and talk to you about this thing. So basically what I want to talk about today is, it's sort of a way to put together a number of things that I've been doing. The talk on Friday is going to be about machine learning type of things, okay? Completely different, orthogonal to this in some non-Euclidean sense. But anyway, so, but basically what I'm going to talk is about some qualitative features that are unique to nonlinear systems, in particular in regards to things like transient behavior and responses to inputs, not classical dynamical systems questions about asymptotics, for example. And you'll see what I mean. And I was motivated by a number of areas of interest. I don't need to say to this audience, but when I talk to biologists, they always say, well, or to other people, that, what does ecology have to do with immunology, for example? How could you be writing papers in all these things, okay? Well, clearly, this is a trivial example of that, right? You've got a predator-prey system, you've got a predator, you have a prey, right? But in the immune system, you may have, say, CD8 plus cells, T cells that are cytotoxic, that attack tumors, and actually they gain from the tumors being there. They get activated by the tumor being there, and they attack the tumor. They eat up the tumor. They don't really eat it up, but you know what I mean. So you also have a predator-prey type of interaction, right? Very similar. No, I'm not going to say the formulas are going to be the same when you write a model, but the basic ideas are very similar. So that's just, again, I don't need to sell you about how mathematics is a universal language, obviously. So I want to emphasize at the beginning why, I think in our community, of course, you're going to jump and say that's not true. I do work that doesn't do that, and we all do. But we tend to talk about stability, asymptotic behavior, and things like that. But transients matter. Okay, so that's sort of something I want to talk a lot about. So I'm not going to talk about this particular work. I actually worked on P53, but not about this particular work. But just to give you an example of how important transients are, there's this gene, the P53 gene. There's a protein that gets expressed, the P53 protein, which is one of the most important proteins in cancer. Half of all human cancers have a mutation in that. Okay, and the only thing I want to talk about is about the fact that when DNA damage happens to some sort, P53 is a sensor that notes this damage and sends a signal downstream to other things, and this signal has been characterized as being of 3 different types. You could have just one pulse, or you could have a periodic pulse, or you could have something that goes to some constant pretty fast. And it turns out that depending on what type of signal happens, you get a different outcome for what the cell is going to do. The cell is going to stop dividing, or the cell is going to die, or it's just going to, try to repair the damage, and so forth. So this transient behavior turns out to be really important in directing the cell what it should do. An example that we are even more used to is the example of epidemics. We all were here, 5 years ago, and let's think about what happens in that case. Suppose you take the standard SIR model. If I have time, I'll later talk a little bit about that, but I think you've all studied that in some class. And actually, it's a very good model. It turns out, amazingly good model, it turns out, even though it's so trivial. Anyway, you take the standard model, and assuming there's no reinfection, let's say once you had infection, at least at that time scale, you don't get infected again, then what's going to happen is the number of infectives eventually is going to go to 0. That's a theorem, okay? It goes to 0 asymptotically. So nothing interesting there. As a matter of fact, the number of people that are susceptible, the people that never got infected, goes to some constant value, non-zero constant value. Those are all theorems you can prove, okay? So that's not terribly interesting. But what's interesting is that during the behavior, during the epidemic, there's a number of people that are infected at any given amount of time. You may have behaviors where you have an L infinity norm, if you want to be fancy, right? A supremum that is very high. And we all heard about flattening the curve. We all heard about how what was really important is not how many people are sick over a month or 2 months. What really matters is that in no one day we have an overwhelming of the medical facilities, right? We have enough respirators, although just respirators were a bad thing to use. But anyway, so ventilators were a bad thing to use. But the important thing was to minimize sort of the transient, right? That's really the important thing. So I actually worked on this, optimal ways to minimize transient. But the one little thing I want to point out, because this is going to happen a lot in the lecture, is there's an interesting sort of motif as a purely, just you guys should write the equations, but just to understand it. Suppose beta is the rate at which things transmit. So for example, this number could be controlled. It's lower if you tell people they all have to stay home, okay? It's higher if you tell people, hey, go out, go to school, and whatever. So as beta increases, if this quantity beta is larger, then of course the number of infectives will be larger, right? Because you get more people getting infected, and the number of susceptibles goes down. But the more susceptibles you have, the more infectives you have. So you have an example, the first one that I'm mentioning here, of something called an incoherent circuit. So you have beta has a negative effect on S has a positive effect on I, so this effect is a negative effect. We'll look at Jacobians of things, right? We'll be more precise later. And beta, on the other hand, is positive on I. So you have these two opposite arrows in some sense. Is it not clear now? Will be, I think, more clear as we go on. So here's another example where you get these kinds of incoherent effects. This is something I've also been working on a lot. It's in the context of cancer chemotherapy. And one thing that happens, so we actually studied with biologists specific experimental work on this and did model fitting and optimal treatments and so on. But what happens in this particular problem that we were studying, in this particular problem, the interest was in the fact that certain kinds of chemotherapy actually will destroy certain parts of the immune system, not the whole immune system, but certain parts of the immune system, cells that divide that divide fast, okay? Because that's what chemotherapy does, is it damages DNA during cell division. So classical chemotherapy. So what happens here? As you apply more chemotherapy, you're actually, the blunt arrow means a negative effect, okay? Sorry, that's terminology from biology. So as you increase the chemotherapy, this is how control you, you're going to decrease the number of immune cells. You're also going to kill the cancer. But the point is the immune cells actually also kill the cancer, right? These certain immune cells. So again, you have this thing where you have this negative, two wrongs make a right? So you have a positive effect this way, you have a negative effect this way. So again, we have one of these incoherent effects, okay? We'll get back to the motif a lot. I sort of gotten obsessed with that. Here's another example where that happened in other work that I was doing also in immunology that had to do with the effect of what are called T-regulatory cells. So these are cells which will downplay the activity of the immune system of T cells that do killings, CD8 cells. And what they'll do is they will damp this immune response. So they say, okay, that's enough. Don't keep going, okay? But what happens is a tumor cell at the same time will lead to a expansion of the cytotoxic T cells, but it also leads to an expansion of the Tregs, but the Tregs have a damping effect on this guy. So again, we have one of these incoherent loops. There's, of course, other arrows here. The T cells also kill the tumor, so I'm ignoring the dotted arrow there, okay? That's a different issue. But again, we have this kind of thing. And actually, I don't know if I'll get time to get to this because it's towards the end, but I could tell you a story about this that I thought it was very interesting. I'm not going to talk about this, but this is when I got started with a lot of this. I was watching this lecture about checkpoint inhibitors, which is one of the most useful and successful immunotherapies. And one thing that was shown in this slide was how, for a particular patient, they actually had stopped the clinical trial because the cancer had progressed, okay? But it turns out that after a long time, the cancer actually went down. But according to the clinical trial protocol, they need to see what happened at 3 months or something, and they said, oh, it was a failure. It turns out eventually it was good. And people understood after a while, and this is in the standard thing that doctors read when you go and they need to do a procedure or something, or need to understand your condition, this thing called UpToDate that they all look at. And it actually is saying there that you're going to have an inverse effect, right? Like in non-minimum phase system or something, okay? So that says that there's some, so the typical thing is apply therapy, you're going to be a response in the wrong direction, eventually will go down. So it tells you there's a second order system going on in there, right? Some interesting dynamics that is transient, okay? So anyway, so we have this incoherent free-forward loops and whatnot. They could be happening inside a cell. This is some of the pathways that become important in what is called checkpoint inhibition, but also among populations of cells, right? So you can have interesting circuits inside cells, different signaling pathways with proteins inside the cell, different enzymes doing different things, or you could have interactions between different things. And immunology is probably the hardest thing you could think of, okay, to study. I mean, I'm beginning to think it's even harder than the brain, but that's, subject of the discussion, but it's a really, really hard subject because there's so many different interactions, so many different players. So let me get a little more mathematical. So I don't need to tell you about this, but for other audiences, you need to say this. Being that I'm a control freak, a control theorist like all of you, I tend to think of not just sets of differential equations in black, but I tend to have, of course, inputs and I tend to have outputs, okay? But you need to tell other people in dynamical systems about those things, right? And I think in this audience, I don't need to talk about inputs and outputs. So that's going to be the formalism. I'm going to talk about, so everything I say is going to be for ordinary differential equation systems with inputs and outputs. The outputs sometimes don't matter, depends on the context. But there are similar things you can do for delayed differential systems, PDEs, whatever adjectives you want to add. You can do similar things to a lot of things. Not all of them, but a lot of the theory will generalize. So the key thing I want to talk about is I want to associate the obvious graph. I'm not going to say anything non-trivial here for you. I'm going to say, okay, if I have a set of differential equations, I'm going to look at the right-hand side, the vector field. I'm going to look at the Jacobian of that. Of course, it depends on the point at which I'm evaluating it, but I'm going to assume the signs of the Jacobians are constant. We can discuss that assumption, okay? But in biology, it's a super reasonable assumption. And I'm going to say that a variable x1 affects positively a variable x2 if the derivative of f2, the second the second entry of the vector field with respect to x1 is positive. Okay. In other words, if increasing x1 leads to a faster growth of x2, similarly, I'm going to say a negative one corresponds to the Jacobian entry being negative. Increase of x1 leads to x3 going down, and of course the same with outputs, with inputs, and the same with outputs. Okay, so it's just the obvious thing. Take Jacobian and look at the signs of the Jacobian entries. Okay, that's nothing. It's as trivial as what you're hearing. There's no secret. Okay, all right. So let me remind you about something that actually goes back to sociology, years 1950 or so. People were interested in these kinds of questions. Suppose that we have that's supposed to be node, not node. I'm sorry. So I will call a spin. So suppose you have a graph that has a directed graph whose edges are labeled plus or minus. Okay, and minus is one of those, blunt arrows. This means a minus. Okay, so a spin assignment to the nodes of the graph, and let's say nodes, a spin assignment is an assignment of plus or minus 1 to each of them. Okay, so instead of plus or minus 1, if you want, call them red and blue. Okay, so if we assign a positive sign to this person and a positive sign to this person, and the arrow here was a positive arrow, we're glad. We say they're consistent. Okay, both of them are living in California. Okay, if we have a positive arrow in one, blue and a red arrows, right? Then that's consistent, right, with this being a negative, okay? Because they have opposite signs, okay? So this is the California person talking to the Texas person. Oh, I'm being recorded. That's bad. Okay, there goes your funding for UCSB. Anyway, all right. So sorry about that. You could edit it out later. Okay. So on the other hand, it's not okay to have a spin assignment with this guy's negative and this guy's positive because this is a positive arrow. It's inconsistent, right? You want one times the sign be equal, the sign of one times the sign be equal to the sign of the other. Okay, so I'll say that an assignment is consistent if every edge in the graph is consistent, and the graph is balanced if there exists a consistent assignment. And in principle, there are 2 to the n possible assignments, but it's easy to check this in linear time with the spanning tree, and you can actually see that the whole thing is equivalent to saying that every undirected loop has to have a positive parity. That's equivalent to saying that there's some spin assignment that is good. So, so why am I telling you this? Because I want to talk a little bit about monotone systems. Now, monotone systems is something that, again, a lot of you in the audience know, but let me remind you for the ones that need some reminder, that if we have a partial order on states, and maybe inputs and outputs, if there's outputs, a monotone system is one that preserves the order with respect to partial order. So, for example, in my examples, the partial order will be what I call the northwest order, northeast order, okay? In the northeast order, looking from your side, this guy is bigger than that guy, because the first corner is bigger than the first corner, the second corner is bigger than the second corner, okay? You're to the northeast of the other guy, the bigger one, okay? All right? Okay, so all I'm saying is if you start with 2 states in that relative position, as you evolve in time, they still have the same order, okay? All right? Okay, so, and if there's the input, same thing. So if you start with an x0 less than z0, like this, they keep the same order. Of course, if they started incomparable, then you don't say anything, because you don't know. There's nothing to check, okay? So that's what monotonicity means. This is a classical theory in mathematics, going back to Steve Smale and Mo Hirsch and definitely Hal Smith, who wrote the beautiful book. What we did a long time ago with David Angeli is we extended the theory to monotone systems with inputs and with outputs, which is sort of a trivial thing when you think about it, but it turns out that that's what enables monotone systems to be really useful, because you can take small systems and start connecting them together and build larger systems that are monotone, or maybe are non-monotone, but there may be a monotone with a negative feedback, as we'll talk a little bit about, and there's this theory that in some sense you could always embed monotone systems in arbitrary systems in sort of systems with twice the dimension that are monotone. I'm not sure that's particularly useful for what I'm going to talk about, but people like Andy have been, I think, thinking about some of those things, right? But maybe not, but some people are, anyway. So why are monotone systems interesting? Because, for example, or balanced net, so the main theorem is that a system is monotone with respect to one of the 2 to the n possible orthants. The cone could be this one, it could be this one, right? And you have 2 to the n orthants in Rn, if and only if the graph is balanced. Okay, so you have an easy way to check monotonicity in terms of the Jacobian. Okay, But of course, there are other cones that are not necessarily polyhedral cones, or they're polyhedral, but they're not one of these special cones, and the theory applies to all of those. But those are the ones which is particularly trivial to choose things. And in some sense, having monotone systems allows you to predict things independently of the details of the dynamics, of the algebraic forms of dynamics. So for example, if I have this guy, which is nice and monotone, a balanced graph, you can see that if I increase 1, that's going to increase 3, it's going to increase 2, and, sorry, did I do it right? And, sorry, you have the opposite. Sorry, did I say that right, or did I have an arrow wrong? Oh, no, right. If you increase 1, 2 will go down because this was a negative arrow, right? If 2 goes down, that has less of an effect in 4, which can then go up, and that happens on this side too. So the bottom line is that if 1 goes up, 4 is going to go up, and it doesn't depend on the relative numbers and everything. So on the other hand, if you have a situation like this, when you get opposite effects, when 1 goes up, you don't know if this guy should go up and down. It depends on the details. And so the good thing about monotone systems is that you can make statements that are sort of independent of the details. All you need is qualitative information. The bad thing about monotone systems is you can do things that are independent of the details, because we know that bifurcations matter, right? And parameters matter often. So it depends on the problem, right? So it's very limited, but when it works, it's really cool. All right. So this is basically what I said. I think I'm not going to, I'm going to skip some of the evidence for monotonicity. I'm going to sort of skip this. I'm just going to give you one proof, because I won't do any proofs in this lecture, but I just wanted a quick proof. Theorem, if you have a system that is monotone with respect to, say, the first orthant, the first quadrant, in the case of it's in the plane, you cannot have any periodic orbits. Proof. Assume that there is a periodic orbit. Suppose it's, first of all, it's counterclockwise. The proof is the same in the clockwise case. Pick 2 points like this. Let them move, okay? Now assume the vector field is Lipschitz, so solutions cannot cross. So at some point, you'll have an x that is as much as possible to the right in here, so the z is going to be on the other side, so they don't have the right order anymore. So it's impossible to have a periodic orbit, end of proof, okay? Same thing if you have a clockwise curve. By the way, Smale proved that you could have periodic orbit in 3 dimensions, periodic orbits in 3-dimensional monotone systems, but what is known is they have to be unstable, okay? So they can never be stable limit cycles. Okay. Now, there's a composition theory that goes to monotone systems. The reason I'm going to, this is actually when we started the work with David Angeli in 2003 or whatever, was in order to understand this. Suppose you have some sort of complicated system, okay? So there's some complicated system, in this case is a gene transcription system. And you know that if you're going to put some input, let's say a constant input, for example, constant, and let's say the system converges to some equilibrium state, and you look at the output, and you write your sort of DC gain, so to speak, which is some function of this constant value of the input, okay? So you have you write this thing, we call it the characteristic of the system, okay? So put an input u, you go to some steady state which depends on u. Let's assume that it's globally asymptotically stable for every input u, there's a steady state xu, and we look at the corresponding output, call that k of u, okay? So for a monotone system, you always get monotonic responses. But now let's assume we ignore what's inside that box, okay? We make it into a black box, and all we have is this information. What can we say about a feedback connection? Okay, what can we say about, so we have this is basically what I just said, but what happens now, I'm going to skip, this was about how you embed into a monotone system. But let's say, suppose that we have a system that is monotone, and we put in a negative feedback, so that destroys monotonicity, okay? And I don't know anything about the inside of these systems. All I know is that both are monotone, but there's sort of a sign reversal, like a negative sign. So I know that I have it's basically the way you formalize this is you say it's monotone with respect to a converse order, but just think of negative feedback. Then, in fact, you can give a very simple theorem that says that if you check, it's sort of like a small gain condition. That's what we called it for monotone systems. If you write the 2 characteristics and you do your standard spider web iteration as if it would be a discrete iteration. If the discrete iteration converges globally to a point, derivative of course has to be magnitude less than 1 near the point, but if it converges asymptotically to a point globally, then that system is globally asymptotically stable, no matter what's inside those boxes, okay? So it gives you a very powerful way of deciding things, and it also works if you have arbitrary delays in the feedback loop. That's an easy extra thing that we did with my student Herman and Ciso. So then we said, well, let's try to apply this to a particular case where obviously this is not going to work because there's actually a Hopf bifurcation in the system. allegedly. So you take a standard book, one of the most classical books in mathematical biology, okay? And there's an example of this testosterone dynamics system. It's written like that. I'm not going to go through the details. But you look at the system, and it's easy to see that this is a monotone system with a very simple feedback. And you can check that condition. It's actually trivial to do, to show that no matter what delays you have in that system, you always have global asymptotic stability. However, in this book, there was a claim that this system has oscillatory solutions. Okay, there was a 10-page proof or so that this system has oscillatory solutions. So we actually proved that that proof is wrong. Okay, it took us much longer to find the mistake in the proof. And if somebody wants to actually look at, without looking at our paper where we explain the mistake, look at the book and try to find the mistake. But anyway, it took 1 second to show that that it was wrong. Okay, so I want to talk a little bit more about these kinds of things. So I'm going to talk about feed-forward and feedback motifs. So a feed-forward motif is one where you don't have any actual loops, directed loops. Sorry? We don't have any. No, apparently no. And I've talked to colleagues who I mentioned this, and they say, oh, I teach that all the time. And I said, well, did you ever do a simulation? No, the proof is in the book, and it's complicated, too many parameters. I never did a simulation. Yes, I know. We're being recorded, so I want to be careful with it. Huh? Anyway, I don't want to, yeah, off the record, now being recorded, we can talk more about this. Some interesting stories. Anyway, so I'm not going to, anyway. All right, so you want to show that no matter the parameters, no matter the delays, it's always globally asymptotically stable. Again, it follows in 2 lines, basically, okay, once you have the theory. Okay, so I'm going to talk about feed-forward loops and feedback loops. And it's obvious, right? If a feedback loop is an incoherent or negative feedback loop, it's as you go around the loop. In this one, as you go around the loop, you get a net negative effect, right? And it's a positive feedback loop. If you go around the loop, you get a net positive effect. And for feed-forward things where the arrows still go, maybe in only one direction and you don't have loops, I'll say that you have a positive or coherent situation where any 2 paths have the same sign, and incoherent if there's 2 paths that have opposite signs. So feed-forward motifs are really important. Many of you have seen how they appear in biology. I will have to skip that. But let me tell you one theorem that we have with David Angeli. So what we prove is the following thing. Suppose you have a system, and you apply an input to the system, and the input is monotonic, increasing or decreasing. Okay? Then you can never have, so input is monotonic, okay? Then if you observe any coordinate that either goes up and down or down and up, okay, which is not monotonic, then necessarily there has to be something inside that system which is either an incoherent feed-forward loop or a negative or incoherent feedback loop, okay? That's a necessity condition, okay? So why is this sort of interesting? So I was talking to some biologists, and there was this interesting pair of papers. It has to do with a particular hypoxia response, lack of oxygen response in tuberculosis. But let me, the biology doesn't matter for the point I want to explain. Our paper has lots of details about the biology. But the point is that there was an experimental paper that showed that with this input, okay, this is a proxy for the input. The input is actually oxygen stress outside, but there's this particular sigma factor, this guy that reacts to that and affects everything under that. And this guy is increasing, okay, clearly in the experiments, even with whatever number of replicas they did, but the response is clear. The response is here some gene expression, okay, of a particular gene is clearly non-monotonic. There was a second paper, okay, so that paper was a PNAS paper. There was a second paper, Journal of Bacteriology, saying, oh, well, we understand how this works. This is basically the diagram that explains the whole thing. Well, guess what? In this diagram, there's no negative feedback loops, okay? Or no negative, or no incoherent feed-forward loops even, okay? So these 2 are inconsistent, okay? The 2 papers are inconsistent. Didn't take us any work. It's just looking at it, okay? So the theory sort of helps you invalidate things, I would say. And we went on to understand, well, are there other things, talking to the biologists, that could be responsible for that? So let me talk a little bit, to continue with this, about the question of sensory adaptation, okay? So the word adaptation in biology is not adaptive control. It's also not in the sense of evolutionary adaptation. It's adaptation in the sensory sense, okay? So the refrigerator goes on, you don't notice when it happens, or you notice when it happens, but then after a while you don't. When it goes off, you notice it happened afterwards, you don't notice anymore. Your clothes you put on, you're not noticing them right now, right? You notice it probably not after a while. I'm speaking very, very boring. you're going to notice, right, when I make a change. But if I'm talking the same, sorry. But if I'm so after a while, you'll probably disconnect and start checking your email, right, or thinking, or So anyway, this happens in every kind of sensory situation, okay? That we have this kinds of adaptation to. We tend to detect changes as opposed to detecting the actual signal itself. So one thing that is true is if you're going to detect a change and then go back to normal, our response to a step change is not going to be monotonic. Okay? So the system has to have inside either a negative feedback loop or an incoherent feed-forward loop. Okay? All right. So far, so good. But how do I know if there's a negative feedback loop or an incoherent feed-forward loop? Is there a way to distinguish between the two? And this gave rise to this other work, which says the following thing. Suppose that you have a system that adapts to constant inputs, but now we test the system with periodic inputs. We're talking about entrainment, right? So this is not going to happen for contractive systems, for example, what I'm going to say, okay? And suppose that when you put a periodic input, some periodic input, you actually observe a response which is subharmonic, okay? So you get a period that is longer than the period that you're forcing with, or the frequency, which is a fraction of the frequency. Okay, then you can conclude, I'm not going to prove the theorem, but you can conclude that in fact, there has to be a negative feedback inside. Feed-forward loops can never have this property. So it gives you a second test, okay? Use periodic inputs, and in fact, this was done. There's a particular odor sensing circuit in worms, and they do a beautiful adaptation over 100, several orders of magnitude concentrations of inputs. They react very well. People sort of understand the circuits very well, but there was a question, is there inside that adaptation, is it due to an incoherent feed-forward loop or negative feedback loop? Well, we can't say for sure, but what you can say is, here's an actual neural measurement, okay, of this neurons. It's through some, I think, calcium release. But in any case, but of course, you use some microfluidic experiment to give it, this other molecule that you're giving it that reacts to. And after after a little transient, but a small transient, a period of 20 seconds gives a response. If you really read the scale, the response are actually 10 times bigger period. Okay, so that suggests that we have a negative feedback loop, because in adaptation said we have either that or feed-forward, negative feed-forward, but negative feed-forward could never give rise to this kind of thing. Of course, biology is more complicated, right? But at least it gives you some idea that this is much more compatible with a negative feedback loop than incoherent feed-forward loop. Okay, so let's talk a little bit about Weber's law. Continue with the same topic. So Weber, father of psychophysics, had discovered this idea that sensing is sort of logarithmic. So what do I mean by that? We detect differences not, we don't compare actual magnitudes, but we sort of compare fractional changes. So for example, if I'm holding something that is a pound and you add 100, let's say a kilo, this pound stuff, I don't understand like we're talking about. Anyway, there's a kilo and you add maybe 100 grams, I may notice it, but if you give me something that's 2 kilos, you may need 200 grams before you notice. It's not perfect, but that's roughly what happens. It's been shown to happen in all these senses, and of course we do, decibel measurements for sound and stuff like that, which is sort of consistent with that idea, right? So basically, there's a logarithmic relation in what we perceive. We perceive differences in logarithms or ratios. That seems to be the most important thing. This is actually important from the point of view of sort of robust perception. So suppose that I have a system, which I'm doing here a very simple model. And if you don't know the biology, it doesn't matter. You can just look at this simple differential equation, okay? But basically, we have this, and I'll tell you why I was interested in this. So we have this system in which there's some external signal. This is a mathematician's cell. Anyway, so there's some external signal that leads to some change inside. An enzyme becomes active that was inactive before, okay? And there could be some nonlinear function how this happens or whatever. What you can see is that independent of the total amount, now this is a reversible reaction, so at the level of signaling, the sum of the two can be assumed to be constant. Okay, I'm not worried about expression of the protein and degradation. So at that level, there's a constant amount P total, let's say. Okay, now it's easy to see that the amount P total, okay, will of course affect the signal. Okay, but it's easy to see that if you look at what the amount of P active in response to a given to a given input, it will scale as a function of time. Whatever it is, it will scale according to a number which is the related to this total protein concentration. So the bottom line, the only thing I want to emphasize is that if you have this kind of situation and you want a behavior here which is independent of the total amount of protein, you have two cells, typically their amount of proteins are going to be different. They're not going to be exactly the same. They could differ by a lot, actually. And however, they should react more or less in the same way. This could be a signal that says cells should divide, or this could be a signal that says cells should die, should grow, whatever. That you would expect that should be the same behavior, no matter how much protein you have. So the question is, how do we build systems that have robustness to this scaling in U? And I'll get back to that in a second. So let me just say that this all came from an experimental paper. When talking about it, it was all having to do this Wnt protein pathway. So I'm not going through the detail, but it's just to say that the amount of this particular protein that you have in the cell doesn't matter what it is for this purpose, but the point is that what you see at the end of the thing with this gene expression, which is actually phenotype, you can actually, it actually is the same for different amounts of proteins that you had. So somehow the system downstream is responding to this scaling of the input in the same way. Okay, I'll get to this because there's a little mathematics in there. Okay, so what I'm saying is this. If we have a system that is perfectly adapting, which, by the way, if it's a linear system, perfectly adapting means that this again is 0, okay? There's a 0 at 0. That's what, right? You want the step response to be 0 for a constant input, okay, for example. Okay, so you have a perfectly adapting system. If I have an input that is stepped like this, you get a response, then you get another response, get another response. But of course, if this was, for instance, a linear system, the response will scale with a U, right? It's linear, so it will scale with U. Okay, but what this suggested is that some responses should only depend on the ratio of the change that you had, okay? So in particular, a system that has what we're going to call changeful detection, I like to call it a scale invariant, this system is going to have the property that if these ratios are the same, okay, then what I'm going to get in my response is going to be exactly the same as the other, the same amplitude, the same duration and everything, okay? Linear systems can never satisfy this, obviously, okay? All right. And this is, another way to say it. So the general way I like to think of this, a paper that we published in Sayat about 13 years ago, is that I might have a group of transformations. For example, scalings, okay? A group of transformations on input values, okay? Call them input symmetries. Let's say a Lie group of transformations that sends an input value into some pi of U, okay? And what I like to say is I like to say that if the initial states were pre-adapted to what you had before, you say you have a periodic signal, but at the beginning you had already adapted to a steady state that corresponding to the corresponding guy, then the response should be exactly the same for those two inputs. I should get the exact same response, okay? How could that ever happen? Well, I'll show you in a minute, but before that, what's the main theorem? The main theorem is the following. Let me define an equivariance, okay, to be just a change of coordinates, basically, okay? So an equivariance is going to be a mapping from the state space into itself, so that if I first apply an input and then map to the new set of coordinates, so to speak, I get the same as first mapping set of coordinates, and then applying pi times U, okay? The corresponding, okay? So for each element pi, for each scaling, I should have a particular row sub pi change of coordinates that changes so that you get the same thing, and everything should be consistent with the output. You can write this, of course, in infinitesimal form as a quasi-linear PDE. This is all fairly trivial that you can write it that way. But what's nice is that there's a theorem that says that a system is invariant with respect to a set of symmetries if and only if there is an equivariance of this type. Okay, so in principle it says you have a certificate, like Lyapunov functions or something. We have a certificate, not Lyapunov functions, but let's say a certificate for equivariance. Okay, all right. Of course, you have to find it. It's not trivial. Like it's not trivial to find Lyapunov functions, but it tells you at least how to think of the problem. One way, like with Lyapunov functions, one way is easy. If there's if there is this equivariance, then of course you're invariant to the change of inputs. But the converse is where it requires a little work. Okay, and we did this in this paper. So what are systems that can produce this kind of behavior? This is an incoherent feed-forward loop. I'm not going to go through the details because I have a lot to talk about. But let me give you some examples. So you can just look at the differential equations if you want, okay? So two-dimensional evolving in the positive XY quadrant, okay? So what you can prove is this is what is called, what we like to call a nonlinear integral feedback system, okay? But let me, because basically y, alpha y minus delta would be the value of y. Think of the x, think of changing coordinates so that the x disappears. If you take, say, a log of x or something. But basically, this will integrate in a nonlinear way because there's an x factor in there, will integrate y, or rather alpha y minus delta. So it's easy to see the only steady states of this are going to be precisely, well, either x equals 0, but let's say if I want to look at the positives, will be y has to be equal to that value, okay? And y is my output, okay? Okay, now x, of course, will change as this happens. It's an integrator, right? It should change, okay? This actually, you can see very easily the equivariance. X goes into px, y goes into y. That's an equivariance. So this actually has a property I told you. Similarly, this system has that property. This system you can prove is not equivariant with respect to any, for scale changes, there's no possible equivariant that would work, okay? So there's no possible equivariance, so it's not scale invariant, and which you can see directly too. What's interesting as a side remark for those of you that like systems biology and like Uri Alon's book, if you look at a textbook in systems biology, typically you have this diagram, okay? And people say, yeah, those behave in a certain way. We understand that. Signal detectors, people tell you, okay? But this is the cool thing. These two have the same diagram, okay? In both cases, x represses y. But what's the difference? In this case, x is degrading y, okay? X is degrading y, right? In the second picture, x is inhibiting the formation of y. You have a u over x. X is inhibiting the positive term. In both cases, x has a negative effect on y, but they're different. And even though that picture, that diagram in there was the same, when you actually look at the differential equations and you understand how you interpret that in terms of differential equations very carefully, you see that one of those happens to be scale invariant, the other one is not. So it's completely, experimentally, they will behave very differently, for example, okay? All right, so do we want to do this? Okay, but just to be honest, for the theorem I told you, you want the Lie algebra rank condition to hold, you want the system to be observable in the sense of the classical Lie algebra observability condition. You want that the vector space be real analytic. I don't know how to prove this in the C infinity case, for example. So there's a lot of little, epsilons and deltas, but I don't want to bore you with that. Okay, so reason we got interested in this was because of that paper I told you before, but also because of chemotaxis. Chemotaxis, as you know, is this motion that individuals do, say bacteria, for example, in trying to go up a gradient, okay? The gradient being where the food is. So there's some food distribution, maybe the smells going around, and you smell that the pizza should be over there, so you like to go in the direction of, right, that particular food, and you do this through, in the case of E. coli bacteria, it's well understood, it's the stochastic gradient search, okay? It's very, very well understood. But it's something that, chemotaxis appears in everything from angiogenesis, metastatic invasion. In the case of humans, it appears in cyclic AMP signaling in the slime molds. Just a little thing, because this was sort of cool the first time I saw it. This is, of course, a diagram, okay, a technical diagram of what the different parts of the flagella that make this, the little things that make this thing move, right? The flagella, which are in the back that you see there. But this is an actual picture, okay? This is not an imaginary diagram. This is an actual micrograph picture of what the motor looks like. Isn't that amazing? Okay? There was actually a book about intelligent design saying, with this in the cover, saying this proves that God exists, okay? Anyway, no passing judgment, if you're saying that's what the book said. This was in the cover of the book. Okay, so how do these things work? Well, there's a very like I said, there's complicated things. I'm not going to go through the details. There's some stochastic processes, but in the end, you basically do a gradient, sort of a noisy gradient search towards the direction, towards the maximum of this potential of food, so to speak. Okay. Well, it is some nutrient, okay? People understand very well the pathway, and in fact, you can validate, they're validated models of this pathway. It's a beautiful little system, and you can prove that this has for ligand levels, things react for things over several orders of magnitude of levels of inputs. And you can actually show that there's an equivariance in here, okay? We had predicted this in a paper that experimentally people should know this behavior. This is the actual equivariance in terms of the variables of the system, okay? There's this logarithm thing, but anyway, so we predicted that, and within a year, there was a paper actually validating this. They did an experiment with E. coli. This is a tethered experiment, if you know what I'm talking about. Ligands were changed over huge orders of 2 orders or 3 orders of magnitude, whatever this is, okay, at least 2. And this is when you have this was the input, okay? And the actual response as measured through this FRET thing, I can tell you about if you're interested later where it is, but basically you're measuring something that is a proxy for what it would move. They're tethered, so they can move, but they see how much this flagella are moving. But it's not even that. You actually look at the binding and unbinding to the motor of a particular protein. But anyway, you get that the signals, at least to the eyeball metric, they look exactly the same, okay? And here's another example, maybe not as interesting because it's not orders of magnitude, but here you have 3 signals. This is done in a microfluidic setup, 3 signals that are periodic, and the outputs are basically the same. Of course, it's noisy because it's bacteria, right? But so that sort of was nicely validated, which was sort of nice. We did this also for Rhodobacter, which is a more complicated thing, much more complicated system, also proved FCD. We predicted that. We had a back-to-back paper with a group at Oxford that actually showed experimentally that, within, if you look at it a little bit, from the side, it looks like it validated our predictions. It's not perfect. This is all biology, right? So we got really excited. I remember with my student, my esoteric, we said, well, here's a paper in dictyostelium. We actually predicted that you would have also this fold change detection or scale invariance. So we asked them to run that experiment because they hadn't done that experiment in the paper they had parameters, they had everything. And they said, no, it didn't work. Conclusion, the math is okay, all right? So this paper produced a model with parameters and everything. They had fit to the data. They produced a model. We made a prediction of something that would happen. It did not happen. Conclusion, the model was wrong. The math is right, okay? But it's okay. But I'm saying it's interesting. It gives you immediately an easy test because it's not go and measure this to exactly whatever. You're just saying, okay, try twice. Just put the same signal like twice as big. Do you see something that is more or less the same? Mathematically, it should be exactly the same, let's say. But do you see something that is more or less the same? No, it was very different. So it means you don't have this behavior, right? Which was predicted by the math based on the equations, okay? So, yes?
Question.
Jeff.
So when you have a continuous symmetry, often there's a conservation of odds.
Yes.
Here you have a continuous symmetry, a scale invariance. Is there any...
Well, in a sense, those equivariances are like conservation laws. Let's talk offline about that. It's a good question. Is there another theorem or something? I know where you're leading. Yeah, let's talk offline. Because actually let's talk offline about that. It's a good question. Yeah. So you could also ask about these things are really stochastic. So you could ask about what about stochastic, a stochastic model. You could write a stochastic model. We did this in a paper. I think we published it. I forget if maybe it was an archive, but we never published it. But you can write a little Fokker-Planck equation for how this thing would behave stochastically. If the control system itself has this scale invariant property, you can prove that the distributions are going to be the same. We put different inputs. And this was also, well, we didn't have the complete proof in that paper. We had a word explanation of this in the 2010 paper. And then also the next year, there was experiments done by a group which showed, basically, these are probability, if you like, think of concentrations, right? Along a tube, a microfluidics setup that is there, at different concentrate over time. Okay, this distribution is changing over time. You're putting a nutrient on one side, no nutrient on the other. The bacteria are moving in a certain direction. You start in some initial state, and you see that the populations are changing. They're going. I guess the nutrient was here, so they like to be in this area. Of course, there's some stragglers that didn't make it, but the point is, over several orders of magnitude of concentrations difference, you actually have that the graphs look all the same. Okay, so again, it said the PDE solution should also be the stochastic thing that you can write as a PDE solution. It's also invariant, right? Which is what we had predicted. So much for that. Let me tell you about something. We just submitted this paper. Actually, we just got the reviews. This is something that started with it's again a question of transient phenomena. So it started with some problems also in immunology that I'm fairly interested in, and. Suppose that we have, and this is just a cartoon, suppose that we have some system in which the outputs asymptotically go to the same value, okay? no matter what you do, the outputs, maybe what initial state you start, or in this case, the initial state is the same. We have different constant inputs that we apply. Okay, so for any of these constant inputs, asymptotically, you go to the same value. Okay, so it's a perfectly adapting system. DC gain of 0 if it was a linear system. This is not linear, but okay. All right. Now that's not too interesting mathematically. Okay, but then we ask, what about if we look up to some time t? And you'll see in a minute why that's important. Okay, so we only ask what happens at some time t. And we look at the output up to time t, but what I'm going to do is I'm going to look at the integral under the curve up to time t. You'll see what I'm getting in a second. So for example, for the blue curve, the integral under the curve would be the area of this blue region, okay? And I'm going to plot as a function of the magnitude of the input, scalar positive input, okay, between 0 and infinity. As a function of that input, I'm going to plot that area, okay, under the curve. I'm going to call that the cumulative dose response. Cumulative because it's an integral. Dose response because in pharmacology, for example, dose response means what response you get to a certain dose of a medication, for example. Okay, so a certain input size. Okay, all right. So the definitions are there. Okay. So what's surprising, it looks unintuitive, and I don't have time to go through an example right now of this. Well, the example I'll give you, but it won't tell you anything. It could be that the actual dose response is not monotonic, but the cumulative dose response is monotonic. Okay, actually, I had a picture here with the red and the blue. Okay, so in the red and the blue, the area under the curve, I think, of the red is more than the area under the curve of the white one, or the other way around. Never mind. I forget now what I did in this particular picture. Sorry about that. I have to remind myself. Anyway, so why was I interested in this? So did I have it on the previous? No. Okay. So yeah.
Can you go back to the one with the areas under the curve?
Yeah, I probably messed up the.
It looks like the area under the curve for the blue curve is less than the area under the curve for the red curve.
Yeah.
Which is less than the area under the curve for the green curve.
Yeah, that's probably true. Yeah, yeah, but that's okay. I was trying to illustrate that it's not that you could have one. You could have that. Oh, you see, you could look at. Sorry. Yeah. Thank you, Eric. Sorry. So if you look at the dose response at time t for the red guy, I now remember what I want to show here. Thank you. If you look at dose response at time t, meaning what I observe at time t, how much drug is left if this was a drug problem or something, the red guy has a lower value at time t than the blue guy. However, the area under the curve, if I did it right, in this blue guy is less than the area under the curve for the red guy. I think this, I didn't write it because it was going to be hard to show the shading, but this is a small enough part that compensates, okay? So what I'm saying is not intuitive. Maybe y of t is actually, for a fixed t, y of t could be smaller for the larger dose or smaller dose, whatever, and the opposite could happen for the cumulative one, for the area, okay? It's sort of, when you think about it, it's obvious. At the beginning, it sounds very unintuitive, but it's sort of obvious. Okay, so why was I interested in this? I was interested in this because we were studying the response, or the biologists were studying a response of immune cells to stimulation. They send some signals out, some cytokines, right, to recruit other similar cells to stimulation by when they see cancer cells of a certain type, okay? So the question was how to, what kind of response do you see? Now, what they could measure experimentally, I think I have it in the next, yeah. When they measured experimentally, all they could measure was the cumulative amount of that cytokine that was released. The cytokine is in the dish, wherever you're doing these experiments, the wells, and you measure the total amount at the end of the experiment, okay? How much there is. So you're only measuring the cumulative dose response, okay? And these are actually experimental results. These are real experimental results. 3 sets of experiments by doing the same thing, where you're measuring the dose response, the cumulative dose response, at several times, at 1 hour of the experiment, 2 hours of the experiment, up to 8 hours of the experiment, okay? And you clearly see, at least for enough hours, that you have a highly non-monotonic cumulative dose response, okay? It's biology, so you don't get exactly the same pictures, right? But they're close enough. In the paper, what we show is there's like superpositions of those things with error bars. But it's easier to see them separately, okay, to see how they work. Okay, you can actually take the numerical derivative of that if you want, and you can just see that you actually get some sort of nice even fold change detection, at least for smaller scale invariance, at least for large enough time so you don't pay attention to the small guys, you see that you get almost the same curves, very similar curves. So that suggests that some of these incoherent feed-forward loops, negative feedback loops could be responsible. So, okay, so then, these are the things I showed you before, so I'm not going to go through them again. These are the loops, the different loops I showed you before. I was telling you the CDR may be monotonic, even the dose response is not. This is examples of that, okay? I'm not going to go through that example, but what I want to say, just very quickly, what I want to say is that you can actually prove as a theorem that the first of the feed-forward loops and the integral feedback one, okay, so IFFL1 and IFB, those two actually have a non-monotonic cumulative dose response, okay? While this guy in the middle actually has a monotonic dose response, okay? And the reason that, sorry, let me see if I have it here. What I'm going to do is I'm going to show you the easy one, which is the one for IFFL2. So I think I said it. Why is IFF? Sorry, I said it wrong. I'm sorry. Let me just go back to that. I was going too fast, and I think I have it. Okay, sorry. I said it too fast. Okay, what we can show is that that the CDR is monotonic for the first incoherent feed-forward loop. It's also monotonic for the second one. And actually, you can find examples on with parameters where it's not for the integral feedback. I said it wrong a second ago, okay? All right. Now, for one of them, for the IFL2, it's actually easy to prove directly using this equivariance result. Basically, you can do is, and this goes back to the question that Jeff was asking about invariance. So because of that one parameter group of symmetries, you know that basically, if you scale right in this way, then u divided by x is an invariant, okay? So when you do that, you actually, that allows you to consider a sort of a change of variables that reduces it turns out to a monotone system. In the general case, when you have something a little more complicated with this non-zero k in here, and if you work in biology, you know why I'm doing that kind of Michaelis-Menten kind of thing, you don't quite get in the same argument, but you can still map it into a system that is monotone, so you can conclude monotonicity of the dose response, and therefore also, if the dose response is monotonic, the integral dose response also, the cumulative dose response also is, okay? Not the other way around. Okay, so in that case, it's easy to prove. To do it for IFFL1 was actually very hard. And I would say here, if somebody, you can look at a preprint, wait until we submit the revised version and it gets accepted. After that, if you can show me a simpler proof, I'll be very happy to see it, but not before, because we worked really hard on this complicated proof, so, okay? But I'd love to see a simple proof. It's really complicated right now of why the IFFL1 always gives a cumulative dose response, which is monotonic. So what that says is that one cannot explain the experimental results, okay? Francesco?
It's one of those 7-page proof without simulation.
There's a lot of simulations too. There's a lot of simulations as well. The proof is correct, and there's a lot of simulations as well. Actually, we did the simulations to make sure we weren't making this a mistake, right? Anyway, so we also found integral feedback can indeed give it's a candidate for one of this that could explain the data. The reason so I think I had it here. Sorry. I forgot now. I had a little, I'm sorry. So what I didn't say because it was at the bottom, the screen is usually high and then I can see things. This was low, so I forgot, okay? In that paper, right, we actually said, based on numerical experimentation, that IFFLs did not seem to be consistent with having this kind of monotone response. But there wasn't any proof there. It was just simulations, okay? And there was also an idea of a very complicated circuit that might do it. What's interesting here is, A, we can prove rigorously that indeed that's the case, that IFFLs cannot do this, get a non-monotonic response. And number two, we're actually suggesting that just a simple nonlinear integral feedback could work. Okay, so that's sort of good to know. Okay, I'll probably have to start talking faster now because this is only half of the talk. But I think that Francesco is starting to get nervous. So let me see what I should do. Okay, wrap up? All right, so I'll skip the last 3 parts of the lecture. You'll be begging me to, it's all right. It's like, blame him anyway. Okay, all right, so so I'll be happy to then, let me just tell you what's in those other parts. I was talking about the application of Fenny-Shell geometric singular perturbation theory to understand also something about behaviors of, in this case, since he's just moved out, networks are important in metastasis, okay? The mesenchymal transition and so forth, epithelial mesenchymal transition, and so I'm going to skip that. I'm going to tell you anything about that. And then this part, the epidemics I didn't get to, unfortunately, interesting control problems. I can tell you open problems. If somebody's interested, I can tell you an open problem that I mentioned in that part of the lecture. Unfortunately, Francesco doesn't let me. Or you can ask Francesco, ask us as a question. Anyway, and then, but the thing that was sort of cool here is that I had designed this little idea of how this regulatory cells could interact with this cytotoxic cells, and basically reached some conclusions that were consistent with some papers, which I'm not going to tell you about, that were back in the experimental literature. Basically, this paper said that they observed experimentally that when they took this negative loop in the middle, this control thing in the middle, if they took it out, they got a monotonic response. When they had it there, they had a non-monotonic response. So, and this is a completely forgotten paper. Nobody ever mentions it. I found it once I had the result mathematically. I said, somebody has to have thought of this. And I found this paper, which was long time ago, 1981, okay? I said I won't tell you about all that. So, okay. So thank you so much for your attention and questions. Let me just, I know, I had something, sorry. Go ahead, ask the question. So I just want to put sorry. Oops. Yeah, I wanted to have so the QR code that you can click for,
What did you discover on the epidemic lockdown?
Thank you. So this was a question, right?
Seriously.
Take the SIR model. This is a paper I published, okay? Take the SIR model and ask the following question. Suppose that I'm giving a budget by the authorities. During the next year, I can only have 30 days of lockdown total. Okay? People are there's going to be a revolution if we do more than that, okay? So the question is, what is the optimal time at which I should do it? And I gave a very precise rule for the optimal time, many times, because many lockdowns, okay, how they're separated, and it's a feedback law. But the thing is, the reason that I'm not very satisfied with it is I had to assume that when there's a lockdown, it's complete. Nobody's allowed out of their home. There's zero contact, which is, of course, nonsense because even the people in your house you're contacting. So it's a very idealized situation. When we have, let's say the lockdowns are allowed to be to 30%, okay, of contacts. Maybe you close schools, but you don't close, going to the supermarket or something, right? Then I don't know what the optimal solution is in that case. So the solution I only have in the case of complete lockdowns. We have a paper with Jim Green with a partial result for those partial lockdowns, but it's a very incomplete theorem.
Is the cost function?
Sorry, yeah, right. So the cost function in this case was the sup norm. Hold on. Good question. I think I had it here, right? It's the sup norm. It's the sup norm of the number of infectives. Okay. In other words, again, it's because you want to minimize the load on emergency rooms, right? Yeah. And I think it's a really interesting problem. It shouldn't be so hard.
Follow anything optimal?
Sorry?
Did any government in the world follow anything optimal?
I have no idea, but talking about our community, our friend Ida Saga, right? He was the advisor for the Indian government, and his predictions were amazing. I mean, he was on TV making a prediction exactly in 10 days from now or something where you see the peak. And then that's exactly what happened, right? Using essentially the same model. There was one more variable, but yeah. So I'm amazed about how accurate these things seem to be. And of course, the decision to close, when Boris Johnson decided to lock down in England, it was because of the mathematical model that said there were going to be 500,000 people dead in England. And then sorry?
While partying at 10 down.
Yeah, well, so we're being recorded. Let's not. And then the person that was the president of the United States at that time, I don't want to say the name, he actually read the report that there were going to be 2 million people dead in the United States. And, he said, well, maybe we should do something about it too. So the mathematical models had a tremendous influence in what was done, okay? And they were pretty accurate in some sense. But of course, we don't know because they did the lockdowns. But and of course, in England, it's quite, they're much more, they follow much more the mathematical biologists, the epidemiologists' recommendations. I don't think it's going to happen in this country this time.
Any scientific or
Yeah, scientific.
political question? No, any scientific.
Any questions? Yes.
So earlier you showed a plot that showed a pulse response, and then you showed one an inverted pulse response, and you made the statement that the statement of necessity that there must be an incoherence in the network path to create that pulse.
Yes.
I was just wondering if there's an exception if you have an oscillatory signal that's detained at some set point. Is that also
Exception to what? I'm sorry.
that there has to be an incoherent equilibrium?
So what's the exact conjecture?
Well, you were saying that you need to have an IFFL network motif
Or a negative feedback.
or negative feedback.
In order to, either a negative feedback on an IFFL, you need to have if you're going to have adaptation, if you're going to have a non-monotonic response to a monotonic input signal like a step.
Would
Okay.
auto-regulation be
So,
enough?
but like
So-
an oscillatory network, is an oscillatory network, an oscillatory response count as a non-monotonic response?
Yes,
setting?
absolutely.
That's all.
Yeah.
I was, would auto-regulation be enough to qualify for negative feedback? Just to
Okay, mathematically, no. It's an excellent question. So mathematically, no. Okay, if you have a system x dot equals negative x, let's say, okay, for example, right? It's a scalar system that, by comparison of solutions, if you start with two solutions in an order, they will always stay the same. If you put an input, so let's say x dot is negative x plus u or something, the response will be, it's a linear system anyway, it's going to be proportional to u, right? So you're not going to, if u is increasing, you're going to have an increasing u. You can do comparisons, say for constant inputs, right? You can compare x dot equals negative x plus u with x dot equals negative x plus v, where u is less than v, the right-hand side one is less than the other. By comparison, the solutions will remain in the same order, right? So if they started from the same initial condition, say, right?
Okay.
So, however, that brings up thank you for your question, because that brings up a really important question, which is, if you have a self-loop, in practice, there's probably some delay in there. It's not x dot is negative x. It's x dot equals maybe negative x of time t minus tau or something, right? And then, of course, you could have oscillations, as we know, and all kinds of things, right? So you have to be very, very careful with that. And actually, that's what motivated the singular perturbation work. Because I was giving a lecture about this. It was about 15 years ago. There was a physicist in the audience who asked me, said, well, but what about, because I answered that, and the person said, well, but if the delay is very small, should it matter? So it turns out you can actually do a little singular perturbation kind of study that says that if you have some effects that happen very fast, then, and if when epsilon is 0 in the singular perturbation sense, right, you get a monotone system, then in fact, for epsilon small enough, it's not monotone, but it still satisfies all the properties that you want of monotone systems. It's a very precise statement, a very precise theorem. It was a thesis of my student Liming Wang. It was motivated exactly by somebody asked me the same question that he asked, and I answered, but the person then went forward and said, well, what about if it's a small delay, or what about if it's a small perturbation, right? So, and then we actually were able to prove this, the whole thesis was about this, to prove that you still have what is called global generic convergence. Anyway, I don't want to spend more time with that, but I can tell you afterwards, yeah. So, but yeah, it's an excellent question. So, yeah, self-loops, which is diagonal entries, when you look at monotone systems, don't matter, essentially, okay? So that's the answer to the question. But again, in practice, what does it mean to have a diagonal entry? I mean, are the gradations really instantaneous, or is there a delay? You measure something and then you degrade, So it's a question of modeling, right? So all these things you have to take with a grain of salt, right? Even when I give you one of those diagrams, I mean, they're a little more complicated than just saying this is affecting this directly, right? So my view of these things is we use mathematics to sort of guide what the experimentalists should be trying and give some ideas to the experimentalists. Try this experiment. We predict this is going to happen based on the theory. Maybe it won't happen because the biology was more complicated, right? Than the model shows, of course. So biology is not engineering, okay? It's not high precision, I mean, some areas of biology, maybe, instrumentation, it's okay, right? But if you're dealing with things like cell biology, there's so much uncertainty in everything. You repeat the experiment twice, you're going to get completely different results, sometimes very different, sometimes close enough, like in there. Yeah, ideally they should be the same, but then typically, I recently was listening to a lecture where somebody was saying he took the most common enzymatic reaction measurements, and he asked like 20, 30 labs, 40 labs, I don't know, a large number of labs to do this experiment that would measure the same kinetic rates on these enzymes, and they had results that were all over the place. So, could
All right,
be.
last question.
Yeah.
Madam.
Has any of your past or ongoing work led to new directions for or chemotherapy-induced treatments, or primarily focused on explaining phenomena that are already being
Okay, first of all, yeah, thank you, Madeleine. Okay, thank you. No, that's a great question. I think that especially when you deal with things that are related to medicine, the time constants there are incredible, okay? Things can take so long until something is tried out. So I wouldn't say that any of that had any direct impact. However, actually we did something, but the experiment didn't work out. We said the optimal therapy is this, and it wasn't, it didn't have, maybe our model wasn't good enough. So what I am very interested, I didn't talk about that topic at all, but I am very interested in the generation of resistance to therapy, and in particular, the distinction between Darwinian generation of resistance and what one might call Lamarckian generation of resistance, which people think Lamarckian, Lamarck, we don't, that makes sense, it doesn't happen, but in fact, we're realizing now there is such a thing as Lamarckian adaptation, okay, that happens through things like epigenetic changes, not the giraffe stretching the neck, as we learned in school, but it's more sophisticated things. And then when you have this kind of adaptation, the way you apply therapy becomes very critical, because you're making the cells react by defending themselves. So maybe you don't want to attack them too much because you want to let maybe the immune system have time to. It's not what I was talking about. It's a different thing. Not what I was talking about. Or you might have a population where you have some cells that are sensitive, some are resistant, some are sensitive to the therapy, the others are not. They're competing with each other. The guys that are resistant, they have a metabolic cost to being resistant, so they grow slower. So these guys who are bad, they're cancer cells, they're not as bad as the ones that you cannot kill. So it's good to have a certain amount of those to keep the other ones in check. And now you apply therapy, you kill these guys, now these guys start taking off, and there's no way to kill them, okay? And that's why many therapies fail, okay? So then the interesting question, it's a control theory question, is how do you apply therapy so that you keep enough of the bad guys but not as bad to be there to outcompete the other guys, while maybe the immune system can help or something else can help, okay? And we actually have a lot of models about this and ideas, at least validating. But again, it's explaining for now, but also coming up with, once you have this, you can say, what's the optimal way to do this? We have optimal control type little problems where you say what the optimal way to apply therapy would be in a certain way. What we did not get, it's nowhere near being applied yet, but I'm actually working with people that are looking at things like adaptive therapies, which they're doing clinical trials on. This is not my work, but they're doing this kind of thing. They're trying to decide what's the optimal way to do therapies to deal with this kind of resistance question. And they're very, there's people at the Moffitt Medical Center in Tampa, and they're reporting very good results with patients where they're surviving much longer, based on this, what they call adaptive therapy, which is like sort of feedback control kind of things. Okay, it's not adaptive control, but so there's a lot of use of these models right now, even at the point of doing clinical trials, but not so far. I'd like to, but but no, it's mostly my view is I like to understand, as you said. I like to understand. I like to give some theoretical understanding of things. Or yep.
All right, so let me make a final comment. So you're all invited tomorrow, 3 p.m., 3 to 5, 3:15 to 5, at the Mosher Alumni Hub. We will have a celebration with Eduardo, and in honor of his visit, and of Professor Petar Prokoto, which will possibly join us.
Oh, that would be fantastic.
And so you're invited tomorrow, 3 to 5. And with that, the Mosher Alumni Hub. With that, I would like to thank Eduardo one more time.
Thank you for the invitation again.